We'll go ahead and get started. So it is my pleasure to introduce to everyone here, Harborview, and everyone who's watching from their computers at home, to introduce Dr. Russ Van Gelder, who is Professor of Ophthalmology and Chair of Ophthalmology here at the UW. Dr. Van Gelder began his academic career at Stanford, where he got his BS, his MD, and his Ph.D., and did his graduate work in the molecular biology and pathways relating to circadian clocks. He then went to WashU for residency in ophthalmology, and during that time, during a postdoc, continued to do work on photoreception and circadian rhythms. And between 1999 and 2006, quickly rose along from assistant professor to full professor at WashU. And then in 2008, we were extremely fortunate to have recruited him to the University of Washington to head the Department of Ophthalmology, where he continues to work today. His work, again, continues in the field of non-visual photoreception, but in addition, he's also involved investigating inflammation and the immune response in the eye, work for which he has gotten a Burroughs Wellcome Translational Scientist Award in 2007. And along those lines, he's been funded by the NIH for over 15 years. He is a past president of the American Uveitis Society, the current president of the American Ophthalmological Society. He has published numerous high-impact papers in Science, Neuron, Nature, Genetics. But in addition to his basic research, he continues to commit his efforts to just curing blindness, if I can be that simplistic. And so he'll be talking to us about some of his work along those lines. And on a personal note, I should say that Dr. Weinstein, John Weinstein, and I had the privilege of taking over the space that he had vacated up in the BB 8th Wing, and so we are hoping
this grant
exactly. So we are hoping to get some of that mojo to rub off on us. So anyway, Dr. Van Gelder, thanks so much for joining us today.
Thank you. If I'd known that giving up that space would be so appreciated, we would have done it years ago.
You don't have any more, do you, Russ?
No, that's for sure. But we do have it's slightly nicer space now. So I can get this to stick here. All right. Well, I really appreciate the opportunity to be here and give a very translational grand rounds. Obviously, we're first cousins here in ophthalmology and neurology. So just to remind you, this thing up here exists primarily to serve the eye, which is wonderful, and process the complex information that comes out of the eye. It is pretty amazing that our entire visual world, though, is really just dependent on a series of action potentials coming out of 1.3 million axons in the optic nerves. And so you can get around in the world pretty well on just one eye, and frankly, you get around pretty well on about 500,000 axons. So even people with advanced glaucoma can still function quite well. And it really does take a lot of work of the thalamus and of the, obviously, occipital cortex to decode that signal, and that's kind of going to be the theme I come back to at the end of this talk when we discuss how we might cure blindness by recapitulating those pathways in people who have lost that photoreceptors. So for this audience, I don't need to go into great detail about what the neural processing pathways are of the retina. As you all know, the retina is really the first stop in the brain. It is a sensory tissue, but a neural sensory tissue. It's built backwards. Light actually comes in through the thickness of the retina and has to penetrate the entire retina. So in this image, the lens and the cornea would be down in the basement. To get all the way to the back here to the photoreceptors, the rods and the cones. And the rods and cones are where the magic really happens. That's where we've evolved these photopigments, rhodopsin, conopsin, that convert the energy of single photon absorptions into isomerizations of these chromophores, which then triggers a highly amplified signal cascade through G protein mediated signal transduction through transducin. There are about 100 million rods and cones in your retina, and those really reduce down to somewhere in the order of about 40 or 50 million bipolar cells, horizontal cells, and amacrine cells, which are the processing units in the retina. Quite a bit of pre-processing is done in the retina, and then that, as I said, results in a signal that gets to about 1.3 million retinal ganglion cells, whose axons become the optic nerve and go off primarily to the lateral geniculate, although a small branch of those go up to my favorite place, the suprachiasmatic nucleus, and tell the circadian clock what time it is. So there is a fair amount of processing that actually happens in the retina. Individual ganglion cells can be direction selective, they can be orientation selective, they can have surround fields that are on, they can have surround fields that are off. But ultimately, our whole visual world is compressed into relatively low frequency action potential spikes in this million cells in the ganglion cells. This is the magic that I was alluding to. This is the photocycle, which was worked out beginning by George Wald in the 1940s and 50s, and work still goes on. Jack Sari and Chris Palchevsky, who were here for many years, worked out a good chunk of this pathway. But I want to just remind you that the real magic here is the step where, actually this is the pigment epithelium, this is the outer segment of the rod, so this would be in the retina, is where we go from 11-cis retinal, the aldehyde, to all trans-retinal with the absorption of a photon in the visual spectrum. The exact peak of that spectrum depends on what protein the retinaldehyde is in. If it's in a conopsin, it might be tuned to pick up blue photons or tuned to pick up red photons. Rods pick up a relatively blue-green photon around 505 nanometers. It's a little bit of a cumbersome system in that the chromophore needs to be recycled. So after the photon absorption, this will signal. It's then reduced to an alcohol. The alcohol is transferred to another cell. It's esterified. That ester is then reisomerized as a complex, and then it's reoxidized to the aldehyde, which is then exported again. And the reason I bring this up is that many things can go wrong in this pathway, beginning with the opsin molecule itself being mutated, mutations in lecithin retinal lysyl transferase, mutations in RPE65, and all of these mutations result in genetic forms of blindness, which right now we have no treatment for. Most of these result not only in blindness, but also degeneration of the retina, because a rod or a cone, particularly a rod that doesn't signal, that doesn't have photopigment, is not a happy rod. In mice, if you make a rhodopsin knockout, the outer segments of the rod degenerate. So the absence of this activity apparently is deleterious for rod function. So outer retinal blindness, which basically means loss of the photoreceptors, there are two big flavors of this. There's the hereditary forms and the acquired forms. I'm not going to talk much about the acquired forms, but it's the same type of disease. The acquired form is really age-related macular degeneration, which is the leading cause of blindness in this country and in most of the developed world. Very, very common. Look to your left, look to your right. When you're all 80 years old, one of the three of you is going to have macular degeneration of some level. 15% of people will lose vision from macular degeneration. So this is our senile dementia, Alzheimer's type disease. This is really what we see a ton of in the elderly population. The hereditary forms are retinitis pigmentosa and its cousins, Usher syndrome and others. And these are the most common inherited forms of blindness, much less common. About roughly 1 in 3,000 people has retinitis pigmentosa. So not very rare, but uncommon. And that means there's about 100,000 people in the United States who have this. There's some famous people who have this. Steve Wynn is probably one of the most famous, and Gordon Gund. These tend to hit later in life if the mutations are relatively mild. And so people might not be diagnosed till late teen years or early 20s, and then have a slow, steady decline in vision so that by the time they're in their 40s or 50s, they've lost their central vision. Question?
Isn't macular degeneration age-related genetic?
Well, it's probably, the best studies suggest that about 60 to 70% of the risk of age-related macular degeneration is genetic, and that about half of that genetic risk is attributable to one particular polymorphism in the complement factor H gene. So this is one of the few examples where a genome-wide association study actually yielded something useful, the GWAS, because many of those really come up empty. However, it's not strong enough to be predictive, so we don't recommend routine genetic testing for patients because the majority of people who have the complement factor H high-risk allele still will not get macular degeneration. It's multi-genetic. There's also environmental factors. Smoking is clearly a factor which accelerates macular degeneration. Bright light, some question about that. Some large studies suggest that outdoor lighting does accelerate macular degeneration. Others suggest that there's not much of an association there. It depends on whether you follow Beaver Dam study or the Blue Ridge Eye study or which large cohort was being followed. But yes, there is a genetic component to it. So when we look at a retina that has degenerated from retinitis pigmentosa, it becomes apparent that most of the retina is left intact. So when patients have RP, they lose the rods and cones, and that's the big space that's missing here, where we see the outer segments here. But the inner retina is left intact. So the optic nerve is still intact, still presumably functional. The bipolars and horizontals are still intact. There is rewiring, and Robert Mark at University of Utah has done some beautiful anatomic studies to suggest that there is quite a bit of remodeling that goes on when the photoreceptors disappear. But it's also very clear that the retinal ganglion cells in particular do not die when the photoreceptors die. So the nerve is still functional, it's just not transmitting any photic information. This is what it's like to have retinitis pigmentosa from the patient standpoint. Because most of these diseases are rod-based, and because the rod density is much greater in the peripheral retina than the central retina, which is very cone-dominant, the peripheral retina goes first. So people get a shrinking visual field, typically with a ring scotoma when it's symptomatic. And in the end stage, they'll be down to about a 5-degree visual field or less where the cones are. There is a secondary cone death. What's thought to happen is that the rods help support the pigment epithelium, which supports the recycling of the chromophore. And if the rods die, the RPE, the pigment epithelium dies, and then the cones die secondarily. So the cones die much later, typically in middle age for these patients. We can diagnose this disease very easily with electroretinography. First, the retina usually has a very typical appearance with these bone spicule formation of the retinal pigment epithelium. This is almost pathognomonic for RP. Waxy pallor of the disc, which is, we think, a reactive gliosis potentially. Attenuation of the vasculature. And then when we do electroretinography, it's basically flat. So we see no rod responses and severely attenuated cone responses in late disease, and that really makes the diagnosis. Now, it's a syndromic diagnosis. There are at least 150 mutations that have been associated with RP in at least 40 different genes. So just making an RP diagnosis doesn't tell you what the underlying pathophysiology is. Some forms are dominant, some are X-linked, some are recessive. So the question has been, what do you do for this? And there's been a lot of study which really hasn't yielded much clinically useful information on trying to slow degeneration down. Elliot Burson had a long program of trying to do vitamin A supplementation, hoping that more chromophore would keep the rods alive longer. And that really never yielded anything that was clinically meaningful. You could slow the ERG suppression down a little bit, but it didn't really affect vision or visual field. And there are attempts right now to do in situ gene therapy, and especially for the autosomal dominant forms, to use these molecular scissors to basically try to chop up the mutant gene, but those are quite a ways off. Most of the effort really has been, in the last few years, has been around trying to restore function by making the ganglion cells work again in a light-dependent fashion. And there have been 4 strategies that have all made significant progress in the last few years. I think the most, the greatest progress, at least from a general public standpoint, has been this device. This is the Argus 2, which is an optoelectronic prosthesis. And the way this works is it's implanted on the retina surgically. And it's got a little array here, I'm sorry that the projector doesn't show this very well, of 60 electrodes. And a little, it's an 8x8 array, and 4 of the electrodes are grounds, and the other 60 are stimulating electrodes. And it's hooked up with a little wire that you can't see here that goes to its power supply outside the eye, or on the side of the eye. And basically, when light hits one of those electrodes, it's amplified through the power, and it uses external fields to stimulate the ganglion cells in that area. And it actually works, which is remarkable. So if you put this in a patient who's no light perception blind from end-stage retinitis pigmentosa, they will see phosphines of light in correct areas, because of course the retina always maintains its retinotopic mapping. That is, a spot in this part of the world is always that spot on the retina. And if you put a big bright letter A on the side of a barn, they can actually see that and say that's an A. The problem with this approach is that you're limited in the resolution of those electrodes. The amount of current that you have to inject to stimulate a ganglion cell extracellularly is quite high, creates a lot of heat, and if you pack the electrodes more densely, you fry the retina. No one has figured out a way around this yet. So I think this is a very promising approach in that it has shown us that you can stimulate ganglion cells in these blind patients, create visual phenomena that are useful. But in terms of really restoring function to patients, if we're limited to a few hundred pixels, it's unlikely that activities of daily living are really going to be significantly impacted. You know, we like to see ambulation, for example, as an activity of daily living that could be restored to someone who had a real prosthesis. A lot of hype around stem cells approaches, obviously, as well as in neurology. And there is reason to be optimistic. Tom Ray here is a leader in this field and has managed to differentiate photoreceptors, ganglion cells, amacrine cells out of both IPSs and embryonic stem cells. Working with Jen Chow in our department, they've now transplanted these into macaques. The cells live. They actually put out axons, the ganglion cells do, that home into the nerve. But as all of you can appreciate, it's a few years before we're ready for prime time with this. Lots and lots of technical hurdles, safety issues, and so forth. And I think it will be a while before we see a human clinical trial of just undifferentiated stem cell implantation into an eye with retinitis pigmentosa. There's also been a lot of hype around optogenetics in the research community particularly, but this is now moving into the clinical realm. Optogenetics basically means genetically expressing typically invertebrate opsins. So channel rhodopsin or halorhodopsin, which come from organisms like Chlamydomonas, little algae. And what these are opsins that don't use G protein signaling, they actually directly gate ion channels. So if you genetically express a Chlamydomonas opsin in a neuron, for example, then you can create a sodium conductance when you turn the lights on. And that would cause the cell to fire, which you would think maybe you could then program the activity of that cell. So there is a company that's using work that came out of the Pan lab at Wayne State University, putting channel opsin into ganglion cells and seeing if that will restore vision. And I think it's a promising approach. The challenge clinically has been that the amount of light that you need is very, very bright because there's no signal amplification. You're just one protein, one channel. So you don't get a whole lot of current into the cell for each channel that's expressed. There's also the theoretical risk that as a uveitis specialist I worry about, which is if I start expressing a lot of algae protein in the retina, that maybe we would end up with an immune reaction against that and end up with an autoimmune retinopathy. It's a theoretical risk. It has not been seen in the mouse studies and the rat studies to date, but it remains to be seen in people. So what I'm going to talk about today...
Can I ask you a question?
Yeah, of course, Bruce.
How many pixels do you think I need to just walk around?
Yeah, that's a great question. That gets into the whole question, again, of how much information is encoded in the retina. Now, we know that we can, so I'm not going to do it in a pixel sense, I'm going to do it more in a gross information sense. If we consider a 30 hertz signal in each of 1 million ganglion cells, I think you could ambulate on about 10% of that. So if you had about 100,000 cells activated, and especially if they were in the central visual field, and you could activate them at speeds comparable to what we see, I think that would restore an ambulatory level of vision. So what I'm going to talk about today is the approach. This is the new kid on the block, chemical reanimation. And this is work that we've been doing for about the last 7 years now. And it's sort of a spin on the gene therapy. It's gene therapy, optogenetics without any genetics. It's really using small molecules to try to emulate what we're trying to do with optogenetics for a number of reasons. One, you can do a lot more with small molecules in terms of medicinal chemistry. Two, we don't have the issues with gene therapy, which make it very hard to get into people because of the safety regulations and the FDA issues. Three, it gives people a chance for upgrades. So if you have one chemical that works and we come up with a better chemical, the next time around you can get the better chemical, as opposed to gene therapy, which is one and done. Once you've had your gene therapy, you're finished. But in the short run, our motivation was really we thought we could get this to patients faster because the FDA guidelines and just the whole pathway to translating a small molecule is so well established compared to gene therapy, cell-based therapies, and so forth. So the genius behind this is a guy named Dirk Trauner. And Dirk was an organic chemist at UC Berkeley. He moved a few years ago to the University of Munich. And Dirk is an azobenzene chemist. That's his background. Azobenzenes are the moiety shown here. So it's just like it sounds. It's two benzene rings with an azo double bond nitrogen bond between them. And all of you in the audience, unless you have lived an absolutely organic lifestyle, have probably eaten pounds of this because the common use for azobenzene is food coloring. And these were invented, with all of the dye stuffs in the early 1900s. Azobenzenes were a favorite building block in German organic chemistry. But azobenzenes have a remarkable property, which is that that double nitrogen bond, just like the double bond on the 14 position of retinaldehyde, is photoisomerizable. When a photon of the correct wavelength, in this case with the molecule we'll talk about in a second, the AAQ molecule of 380 nanometers, which is kind of near UV or far UV near blue, hits the molecule, it will isomerize from the trans to the cis form, and 500 nanometers or darkness will re-isomerize it back from cis to trans. So that's a useful property, and the insight that Dirk had that was really genius was to realize that if you appended this onto other molecules, you could put a light switch on a molecule. So if you happen to get lucky and you put this azobenzene on another molecule such that the steric interactions of that molecule are permitted in one orientation and not permitted in the other, then the drug would become light active and only active in one part and not another. One lighting condition and not another. So this was originally done with the tetraethylammonium moiety, TEA, and those of you who have neuroscience background recognize this as an old friend. TEA blocks voltage-gated potassium channels and causes cells to fire. We'll talk about the R group in a minute, but the original molecule that they made was called the spark channel, and MAQ, because it was a maleimide as the R group, and they used the maleimide to bind to a genetically engineered cysteine residue on a Drosophila potassium channel. And what they were able to show was that this molecule would bind the genetically modified channel, and that when they turned the light on, they could, when they turned the blue light on, they could block the channels. When they turned the green light on, it unblocked, and so it was actually functional. So in cartoon form, and I will say this is a very nice cartoon that Rich Kramer at UC Berkeley, my collaborator, made, and it's wrong. So when you see this, what's wrong about it is that we now know that the pore that's blocked by the TEA, there are two TEA sites on the voltage-gated potassium channel, one intracellular side, one extracellular side. Cartoon will show extracellular side. We now think it's the intracellular side that's actually blocked, so just with that caveat. But the basic idea is that a cell depolarizes, which allows TEA into the pore. Originally, when Dirk synthesized the chemical, he put an acrylamide on the end of this, and which you can see here. And the reason for that was that this would be basically a nucleophilic reactant and would cause a covalent bonding of this molecule to the channel. So it would stick there just like those of you who've done affinity labeling of channels. It's the same approach. And then you hit it with the one wavelength of light, and the TEA should flip out, business as usual for the potassium channel. Hit it with the second wavelength, it flips in, and now the channel is blocked and the cell should fire. So the original proof of concept that they did on this was to take a hippocampus and just put it in a dish, and then pour about 300 micromolar of this compound, which is called AAQ, which stands for acrylamide azobenzene quaternary ammonium. So we always have this three-letter abbreviation for these. So this is AAQ. And they put an electrode in one of the cells, or outside one of the cells here, of the hippocampus, and lo and behold, the hippocampus became light sensitive. And I saw Rich Kramer present this work at a FASEB conference in 2006, and it's one of those moments you remember forever. You see this thing, you go, oh my God, I can't believe they did that. So I went up to him afterwards and I said, that's just amazing stuff. Have you tried it on a retina yet? And he said, no, we've talked about it, we haven't really done it yet. And I'm thinking to myself, wow, they've made a great model of light-dependent epilepsy. That's not that useful, but you could really do something useful in a retina with this. So we set up a collaboration, and we've been working together now ever since. The reason that we were set up to collaborate on this, and we were just sort of in the right place at the right time, is that my lab had spent the last 6 or 7 years becoming very adept at this recording technique called multi-electrode array recording. And this was partially invented by Rachel Wong and Marcus Meister when they were at postdocs. The basic approach is it's sort of like that chip I showed you, except that it is a recording chip, not a stimulating chip. So the chips we use have 64 electrodes on them. It looks like a little microchip. Each recording electrode has a little etched line here to the amplifier, which goes out to the amplifier. And we put tissue then face down, ganglion cell side down, on the array and seal it on with a little bit of filter paper. And then we can record from multiple cells for many, many hours simultaneously. And this works remarkably well for the retina. because the spacing of the retinal ganglion cells is very similar to the spacing of the electrodes on the array. Our electrodes are spaced at about 200 microns, and they're about 20 microns across. We were studying the intrinsically photosensitive retinal ganglion cells, the melanopsin cells, and those are spaced almost identical to this, about 200 microns apart, so we could record one cell on each electrode. When we record from all the ganglion cells, we can get about 10 cells on each electrode, and if we study the signature of each action potential, we can sort the cells after the recording and record multiple cells on one electrode. So from one recording now, we can record about 400 or 500 retinal ganglion cells, which in a mouse that only has 30,000 retinal ganglion cells total, is actually a pretty good proportion of all the retinal ganglion cells. So this was just tailor-made for testing these compounds. This is what our data look like, and most of the slides I'll show you in the rest of the talk have this format. These are raster plots, and what we're looking at here are 60 channels of data in the 3 different colors. This is just 3 runs of the same retina with the same stimulus. At the bottom, you see the stimulus train. So this is darkness, and then here are 5 seconds of light, 5 seconds of dark, 5 seconds of light, 5 seconds of dark, and so forth. And each channel here is the recording from one electrode. So in a normal retina, what we see from the ganglion cells is that the majority of the cells have a transient on and transient off firing pattern. You turn the lights on, you get a big burst of activity that lasts about a second, goes away. You turn the lights off, you get a smaller burst of activity of the off activity. This is the retina that we started working with the AAQ. So this is a postnatal day 8 mouse retina. At P8, the ganglion cells have been born, they're making their connections, but they haven't made the intraretinal connections to the rods and cones yet. So there's no light input to these ganglion cells, so they're effectively blind. And to double up the blindness, we crossed this into the melanopsin knockout strain that we've been using for many years. Melanopsin is a photopigment in a small percentage of retinal ganglion cells that's directly photosensitive. We wanted to get rid of those cells, so we had a really blind retina. So you can see even after a 1 minute very bright blue light pulse in this retina, there's no discernible signal. And we could block glutamate receptors, it didn't change anything. If we put high potassium on the tissue, all the cells fire, so it's not like there weren't cells there. So then we took the same retina and we took the AAQ compound at about 300 micromolar and we soaked it in the AAQ for 5 minutes and then repeated the experiment. And what you can see now is are 2 things. First, in the dark, there's actually a moderate suppression of activity. And these little lines here, this is retinal wave activity, which Rachel Wang's lab has studied for many years and is well recognized in young animals that they have these spontaneous waves, which were unmasked by the relative silencing with the AAQ. But you can see that with the same light stimulus now, every ganglion cell is firing. So with a small molecule chemical, ex vivo, we've now restored light sensitivity to retinal ganglion cells in a very crude way. One thing you'll notice is we turn the lights on and the cells started firing, and then they just kept firing, right? So we turned the lights off and it kept going, and that was actually predicted because, as I mentioned, the molecule has to relax in the dark, and AAQ relaxes very slowly. Its half-life for relaxation is about 30 seconds.
So we can't see the timeline very well.
Yeah, I'm sorry,
Those
this
are
is
seconds?
This is 1 minute here of light.
1
So
minute
60
of
seconds here, 120 seconds here.
Got it. And so then the rhythm of these waves is
About every 20 seconds, something like that.
happening.
Yeah, there's a whole, Margot Feller at Berkeley has made a whole career studying these. There's thoughts that these contribute a lot to the patterning of the developing visual system. That is, in an animal before eye opening, these waves are surrogates to show correlated activity in adjacent retinal spots, and that helps axonal pathfinding in the geniculate and then ultimately the cortex. At least that's the theory of why these waves are there. And if you disrupt these waves and put TTX in the retina, you do disrupt axonal pathfinding into the geniculate.
But they don't happen in the absence of input from a photosensitive pigment or something photosensitive.
In adults, they disappear completely.
But in the young ones, you showed us a picture of.
Right. In the young ones, you do see them, and I should say that we actually took steps to suppress them pharmacologically here. So this is actually done under the cholinergic starburst amacrines are critical to these waves. So if you put cholinergic blockade into the retina, you block most of the waves. And what happened here, just getting into levels of detail for the cognoscenti, this unmasked a non-cholinergic wave generating mechanism. So we don't know where those waves come from, but they look a lot like the native waves that are normally cholinergic dependent. So I showed you before that you could terminate, you flip the molecule around with the second wavelength of light. So we did that experiment. And here what we've done is to take so this is retina again, soaked in AAQ compound, exposed to a 1 minute of 385 nanometer blue light. And then we superimposed a 480 nanometer green light or blue-green light on top of that. And what you can see is that remarkably the extra photons don't increase stimulation, they actually turn it off. And the reason is that they're driving the equilibrium back to the non-blocking conformation. So this was really proof to us that this was due to the molecule specifically, because you would never see this in a normal opsin where addition of more photons would actually turn off a signal. But what this also meant, and was very exciting for us, is that we now had a mechanism by which we could temporarily control individual ganglion cell firing just by flipping the lights back and forth. So we're getting closer to our goal here of being able to recapitulate action potential trains in response to visual stimuli, because we now have at least some way of crudely turning these cells on and off with light by using 2 different wavelengths. So we went for broke, and we decided to see whether this had any actual neurologic effects. That is, did the animals see anything when we did this? So we took very blind mice, and these are old retinal degenerate mice. They're at least 6 months old. They have the RD1 mutation, which is phosphodiesterase 6B, and they have melanopsin mutation. So we showed years ago that these mice have no pupillary light response, absolutely none. And we took the same animal then and looked at it before we injected 2 microliters of, in this case, 5 millimolar AAQ into the vitreous cavity, and then we looked an hour after an injection of the molecule into the vitreous cavity. So you'll notice that the injection itself decreases the size of the pupil slightly, but it was tonic. But then I think what you can appreciate here is that that pupil is indeed coming down, and it does it with a strange little hippus, which is actually not unexpected, because if we think about what this the superior colliculus is sort of geared for, it's geared for this transient on effect, and then a rapid decay in the signal, and what we're offering it is a continuously high signal. And so it's not surprising to us that there's an oscillatory component to it, but at the end of 30 seconds we achieved about 60% of a normal pupillary constriction. So to us this was very encouraging, because it suggested not only does this generate retinal ex vivo retinal function, but in vivo that's enough to drive a simple physiology. Using the pupillary light response, we were able to, and the multi-electrode array, we were able to figure out the sensitivity of AAQ. Turns out to be relatively insensitive. The brightness of light that it required to constrict the pupil, or to, in this case, fire the action potentials, was at around 10 to the 15th to 10 to the 16th photons per square centimeter per second. To put that in context, that's outdoor lighting level in lux, which you can't really translate to photons very easily, but it would be on the order of 10,000 lux, which is pretty much outdoor sunlight to light overcast conditions. Turning it off took even more light, that required 10 to the 16th, which is really bright outdoor conditions. So not quite ready for prime time in terms of really restoring vision in a room like this. We looked at the pupillary light response as well. And Jack Saichev, who was a medical student at the time, is now a resident with us, did these experiments. And we were able to show that compared to wild-type animals, the decrease in sensitivity of the pupillary light response was around between 2 and 3 logs. So it's about 100 to 1,000 times less sensitive than natural vision. And the pupillary light response never got to 100%. We couldn't constrict the pupil all the way down. It topped out at about 50% or 60% constriction. But a good start. Joe Nemmergut, who was a postdoc in the lab, did one other behavioral experiment. We know that mice are negatively phototactic. If you put them in a little tube and shine a bright light at one end, they will instinctively turn around and run away from the light. Blind mice don't do this, or at least melanopsin RD double mutant mice don't. So we took those animals, tested them before and after injecting a compound in the eye. And what Joe found is that wild-type animals will turn around within a minute of bright light in 3/4 of the cases. Pre-injection, 20% turned around. Post-injection, 60% turned around. A week later, when the stuff had washed out of the eye, and we showed that it was no longer functional in the eye, none of them turned around. Not great data. I have to say that these are not the most compelling experiments to show that there's visual function restored, but it's consistent. Now, the problems with AAQ for developing it further were several. One, it's very blue shifted. So that 380 nanometer light is okay for mice because their lenses and their corneas are UV transmissive. Ours are not. So we block essentially all wavelengths below 400 nanometers are blocked by your lens, your natural lens. So we really couldn't use this in people because it's already not very sensitive, and it would be much less sensitive in a human. Second, that slow relaxation basically meant that if we were going to use this, we'd have to use the Geordie LaForge things and put on a stimulating array for the residents. That was the second generation Star Trek. That's before your time. But you need some sort of a stimulator that would switch the wavelengths back and forth. And that insensitivity was already a bad starting point. So starting with needing 10 to the 15th, 10 to the 16th photons didn't seem practical. So we went back to Dirk in Better Living Through Chemistry. Dirk derivatized the AAQ, and he did it by different substitutions here at the R end of the molecule. This is that original acrylamide, and what he realized was chemically, if you put electron withdrawing groups on that end, it would redshift the molecule. That's how you redshift the isomerization. And the two that we came up with that we studied fairly extensively were Phenac, which is phenylethylenylene, azobenzene quaternary ammonium, and DNAC, which is diethylamine azobenzene quaternary ammonium. They're almost identical. We did a divide and conquer technique where Rich Kramer's lab studied DNAC, our lab studied Phenac. Our results are almost identical. The only difference between the two is Phenac is less soluble than DNAC. But what I'm going to show you physiologically for Phenac is almost equivalent for DNAC. In the test tube, DNAC and Phenac both redshift quite considerably from AAQ. Here's the AAQ peak at 380. You can see that the DNAC and Phenac both now peak closer to 500 nanometers, which is right in our blue-green spectrum. So this is a much better spectrum. Interestingly, we could never get AAQ to shift to white light. We always had to use blue light to shift it one way and then green light to shift it back again. The mix of spectra just really put it in a state where it was not signaling. But with Phenac and DNAC, white light works quite well. And we think the reason is that both of these, in addition to being redshifted, they relax very, very quickly in the dark, like in microseconds, as opposed to tens of seconds to relax. And that also has to do with the chemistry that I don't understand very well of the electron withdrawing group and how it destabilizes that double bond. But from our standpoint, what it means is that pretty much every photon that hits this drives it forward, which is a good thing.
Russ, I noticed that you've got 5 millimole potassium, which is not normal extracellular potassium.
Correct. So we use that in a lot of our recordings to help the loading of the molecule. It only will, as all potassium channel blockers, only get into the channel when it's open. And so the little bit of potassium helps to cause the cells to fire enough that we get the drug in. It's not necessary, but it speeds up our experiments quite a bit. So this is an example here of a 1.5-year-old very blind mouse retina with white light that is soaked in the AAQ for a grand total, I think, of 5 minutes and then washed out with Ames. The stuff is very lipid soluble, and so it gets into the membranes and stays there, which means that even after one exposure, 6 hours later, ex vivo, the retina is still light sensitive. So that's very promising from a clinical translational standpoint. As I mentioned, it doesn't really photoreverse. So if we do that same experiment of putting a second wavelength on top of the first, here we don't see it turn off to the second wavelength. The photons are additive, which basically means that it's turning off on its own and that every photon that's absorbed drives it into the on direction. So that's a positive thing about this new molecule. When we get way out to 550 and 570 nanometers, which is getting into the yellow and even into the near red, then we start to see a little bit of inhibition. If we start from a dark position and drive it back, we can get a little bit of inhibition with long wavelengths, but it's pretty minimal compared to the positive photo switching that we get from the molecule in the shorter wavelengths. This is just a graph of what it looks like from a sensitivity standpoint. Here we can use about 15 watts per square meter, so that's about a log more sensitive than the AAQ was. And we can use white light, which allows us to use a much broader wavelength distribution. So the net result is that Phenac is probably between 10 and 100 times more sensitive in real world lighting conditions. And we're getting to the point where normal outdoor lighting and even bright indoor lighting would stimulate it. Now, one of the surprises, and Rich Kramer first discovered this in DNAC, and we confirmed it in Phenac, is that remarkably, this stuff doesn't work on wild-type retina. which is bizarre. We'd been so focused on these blind retinas that we went back about a year and a half after we started doing these experiments and took some wild-type retinas. It was really a toxicity study to see what happens if you co-opt a wild-type retina with these molecules, and they don't co-opt, which is crazy. So when we looked before and after the wash-in of the Phenac, we still see that transient on, transient off response and very little difference. And in fact, if you look quantitatively, this is with just DMSO, which is our vehicle. This is with Phenac, and those curves are identical. I mean, the retinas look identical with and without the molecule. So Rich has done a fair amount of work on this, and his conclusion is that it's a twofold effect. The degeneration of the retina seems to do two things that are useful for the molecule. One is it seems to activate P2X channels, which let big molecules into cells. And I'll show you some data in a second to suggest one of the issues in the wild-type retina is probably the XAQ, Phenac, DNAC molecules are not getting inside the cell. And the other is that there's an upregulation of a particular class of potassium channels called the HCN channels. And that upregulation, that may be the major channel that's targeted by DNAC and Phenac. So again, this is just good serendipity for us because it would suggest if someone is partially degenerated, that the parts of the retina that are still working wouldn't be messed up by the molecule, but that it might still work in the areas that are degenerated. So that's a plus for us that worked out well. Not everything works out well, and I'll show you that too. We did a little bit more sophisticated visual analyses, although I'm still not happy with this assay. The challenge here is how do you tell that a mouse got its vision back, a blind mouse? You can't do, it's the flowers for Algernon effect. You can't really do a trained assay very easily because you're going to have a few hours of vision. So if you want to do a paired stimulus and teach it to recognize a picture and hit a lever or something like that, you can't do that in an afternoon. And so it's very difficult to devise an assay that really has high sensitivity for visual. So you need something innate visual. We went back to an ancient assay that was described in the 1940s, which is the visual cliff. And this has been used in babies and other, it's an innate response that if you feel you're going over an edge, you back off and you stay on a safe floor. So we set this up with the mice, and the wild-type mice will spend about 70 to 80% of their time on the checkerboard side. So there's a lucite cage where it looks to the mouse like it's going to drop off the edge, but it's just lucite underneath it. The blind mice just go all over the cage. They can't appreciate the edge. But a subset of the animals, not all of them, but a subset of the animals after a Phenac injection in the eye showed a strong preference for staying on the cliff side and not going over the edge. Again, I don't like these experiments. We're still struggling to find a good visual assay to show that we've restored real meaningful visual function. We may not be able to do it in rodents. So on to macaques. We decided to go for broke and see what happened in a primate. The reason for choosing macaques is that their retinas are indistinguishable from ours. They're cone dominant, they're foveated. If I showed you a histologic section of a macaque retina, you couldn't tell me whether it was human or whether it was a macaque. We were mostly interested in looking at the midget and parasol ganglion cells, which make up about 90% of the signaling ganglion cells in that retina. And so we figured if it's going to work, it's got to work in those cell types. So Mike Mnookin, who's now an assistant professor in our department and an excellent patch clamper, used tissue from the Western Primate Research Center. And first we did multi-electrode array, and we saw nothing, which now doesn't surprise us. Now, why don't we see light responses here? The answer is probably that the piece of retina that they were working with was extremely bleached. So this was a cone-dominant section of the retina that had been handled in bright light. There's no pigment epithelium, and so our guess is that this is just bleached tissue. But that should be ideal for showing photoreactivation, no photoreactivation. But this is wild-type retina. So again, that fits with the model that no degeneration, no drug activity. So Mike decided to do the experiment of patch clamping these cells, but put the drug into the cell directly through the patch pipette. And when he did that with 100 micromolar Phenac and used blue light to record, this is what the cells look like, and these are now active cells. So this is in a prep where he has retained photosensitivity of the cells. This is an off parasol cell. You can see that the light pulses between 0.5 and 1 seconds. No response here. In fact, it suppresses any response. Here's a second run of the same cell. And here's after the Phenac has been put in the pipette. And you can see it's reversed polarity now and gone from being an off cell to being an on cell. And same for the second here. And you saw the same thing in the midget cells. This is an off midget. And that converts now to on cells with the Phenac in the pipette. So it means that these compounds can work on mammalian potassium channels, which is terrific. It also means that in wild-type retina, it's not getting into the cells, because if we force it in through the pipette, we get light sensitivity. If we just let it get in by itself, nothing happens.
Russ, the change in the baseline activity is not anything we should pay attention to?
No, that's the drift over the recording time, because these take a few hours and the cells tend to speed up with recording, or at least that's what Mike tells me. But no, I don't think that's anything to do with the actual compound, because you saw that actually here as well, even though nothing happened between the two epochs. So why aren't we moving ahead with Phenac into people? And this is the answer. When we inject this into the eye, and these are cadaveric macaque eyes here that we took the retinas out of, it's so insoluble it forms bubble gum. So you inject this into the eye and it just forms a sludge, and 24 hours later that sludge is still sitting there. This is not something I want to inject into my patient's eyes. You can't control where it goes, you can't control how much drug is coming out of it. This is a real problem. So, Better Living Through Chemistry Part 2. Went back to Dirk and said, Phenac's looking great. We love the red shift. We love the dark relaxation. We love the sensitivity increase. Can you make it more soluble? And Dirk had the idea that maybe it was time to give up the quaternary ammonium, because that charged nitrogen at one end plus all those benzene rings basically makes soap, right? Polar at one end, nonpolar at the other. And so he took the charge off of the nitrogens and left them as tertiary rather than quaternary ammonia, realizing that at physiologic pH, these are going to get reprotonated and again become quaternary ammonia. So the new compound is called red dad. So it's diethylamine, azobenzene diethylamine, and it's red because it's redshifted. That second little nitrogen there redshifts it. Again, very small molecule. Molecular weight is about 400. Extremely soluble. So total success here. What we're looking at is Phenac and red dad mixed with monkey vitreous in an Eppendorf tube. So you can see all this kind of egg drop soup look to the compound in the monkey vitreous for Phenac, and with the red dad it just diffuses through the liquid component completely. We can also make this in very concentrated solutions that we could not make Phenac without it crashing out. We can get it up to 25 millimolar in solution, so it's much orders of magnitude more soluble. Does it work, was the question. So Laura Laprelle was a grad student from Dirk Trauner's lab who came over for 6 months to work in my lab on this compound. This is her recording from a melanopsin RD double mutant animal, and you can see it photoswitches beautifully. We switched MEA systems here, by the way, so that's why our graphs look a little bit different. We upgraded. But the thing that she saw, which I didn't see initially, was that in addition to the sustained activity, you got this very strong on transient with this molecule, and you got an off transient. There, and here. We had not seen that before. So now we're getting something that looks a lot more like native retinal function. And I'll show you why in a moment this happens. It is more potent because of its solubility, whereas we have to use 300 micromolar Phenac to get photoswitching. We get really nice photoswitching at 100 micromolar with the red dad. It works down to about 10 watts per square meter, which puts it a little bit more sensitive than Phenac. We were able to do the experiment now where we inject this into the vitreous. It spreads everywhere in the eye. If we then sacrifice a mouse, put it on an array and measure with 5 millimolar injection, which in the volume of the vitreous, 1 microliter, 5 millimolar, will end up being about a 200 to 300 micromolar on the retinal surface. If we take that retina out, it's now photoswitched. So this blind retina after an eye injection is now photoactive, so that's fantastic. We were not able to do that with Phenac. The action spectrum looks excellent, so it's sensitive in the UV blue out to 480. It kind of tails off around 530 and 550, just like Phenac does, and it's quite sensitive to white light, so that worked well also. Similar to Phenac and Dnac, doesn't do much to wild-type retina. So the only thing it does do, which you'll notice, is it drops the sensitivity of the wild-type retina about tenfold. And I'll come back to why we think that is in a moment. But same thing, it doesn't get into the cells very well if they're not degenerated.
better signal to noise.
Better signal to noise, yep. So here's the actual sensitivity curves. Again, the red dead will stimulate in a melanopsin minus blind retina at about 10 to the 13th to low 10 to the 14th photons if we're using blue light. Interestingly, we did see a drop-off between these two. If you look at these two curves, these are both wild type effectively. One with the red dead here, without here. You can see, especially to blue light, there's about a 10-fold drop-off in the sensitivity, which is what you saw in that previous graph at the 0.2 watts per square meter. And I'll come back at the very end to why we think that is. Now, one of the most interesting things we did here was we pharmacologically blocked the inner retina of these blind animals. And remarkably, when we did this, the drug stopped working, or the compound stopped working. Okay? So if you block glutamatergic transmission with, I think, with your DNQX and AP4, what you're doing is you're blocking any input into the ganglion cells. The ganglion cells are almost exclusively glutamatergically driven. And you would expect if the drug were working on the ganglion cells, nothing should happen because it should still be fine. But in fact, it blocked all of the activity. This is not what happens with Phenac. If you do this with Phenac, you still see activity under blockade. So it suggested to us that the target cell for this compound is different in the red dead because of its solubility or its physiologic characteristics. So we went back to patch clamping. And again, Mike Mnuchin helped us out here. This is a gratuitous picture of one of his fills at the end of a case. It's just kind of pretty. At least on a projector that can show it, it's pretty. But this was a bistratified ganglion cell. And what he showed was that if you took this cell, first if you just put a light pulse on this cell without putting any drug on, nothing happened. If you applied red dead, again in the pipette, or actually this is in the mouse, so he applied this extracellularly, you got light activity. So we're in the mouse now, patch clamped. But if you put the glutamatergic blockade in, you got no activity, which is just what I showed you on the MEA, but at the single cell level. But what he was able to do was then go into the cells and look at whether the currents coming into them were excitatory or were inhibitory. And what he was able to show was that under red dead, if he's here in voltage clamp recording, he could show both excitatory currents coming into these cells and inhibitory currents that were generated after the red dead. So what does this mean? It means red dead is acting on the cells upstream from the ganglion cells. And the ganglion cells are firing as a result of synaptic input from these interneurons, from bipolars, amacrins, horizontals. We don't know which of those cells yet. We have to patch onto bipolars now to figure that out. But this is the second time that serendipity has really favored us, because the farther upstream we get in the pathway, the more signal processing we recapitulate. Ganglion cells themselves, you know, it's on-off basically with this compound, but if you're in the bipolars, you might get direction selectivity, you might get orientation selectivity. A lot of the circuitry is still intact, so we're very enthused about this possibility. In contrast, Phenac, which I showed you before, if we put it under blockade, there's no excitatory currents, no inhibitory currents that are, I'm sorry, this isn't under blockade, but if we record the currents coming into a Phenac activated cell, we don't see input from the outer retina. So Phenac is acting on the ganglion cells, red dead is acting upstream. So behavior on red dead, pupillary light response.
Why it's not acting on ganglion cells?
We don't know. We don't know. It can. If we put it in the patch pipette, we can make it happen. But our guess is it has something to do with the solubility and what cells it's getting into. But I don't know that. I wish we did know that. Pupillary light response, nothing. We injected this into the vitreous, saw nothing. Open field behavior. We put the compound in, put the animals under a bright light. We expect them, DNAC, for example, will cause the animals to become more active in the light. Nothing. We took the retina out of the animal that had nothing and put it under the multi-electrode array, and it was fully photoswitched. So the retina was light sensitive, no effect on behavior. So now serendipity is not so kind to us. We did a light dark box test under very, very bright light. This is just a 2-chamber device that one of my technicians built. It basically has a little pipe there. The animal can either hang out on the dark side of the box or the light side of the box. If you do this in blind animals, they will spend just about exactly 50% of their time on either side of the box. Wild type animals spend less than 20% of their time in the light side of the box. They really like to be in the dark side. When we do this with the blind animals pre-injection, whether we keep the box in the dark or the light, about 50% of their activity on either side randomly. If we inject with the red dad, finally we get a little effect, which is that they actually prefer the light side of the box, paradoxically. We think that's what we call positive masking. I don't have time to go into this in great detail, but Nick Murzowski at Toronto described this years ago in animals that have very little vision. Under very, very dim light conditions, animals are actually not negatively phototactic, they're actually positively phototactic under very, very dim light. So we interpret this as meaning that the animals perceive a very, very dim light. So what's going on? Why did we get such great in vitro results and then see nothing in vivo? And this is what we think is happening, is that we've been hoisted by our own petard. The solubility of this stuff, when you inject it into the eye, it goes everywhere and it stains everything. And it stains it bright yellow. When you dissect these eyes out, this is the lens of one of these mice. And you can see that lens looks like an old person's cataract. It is totally yellow. Here it is, normal lens, and after injection of Phenac into the eye. So we calculated, based on the molar extinction coefficient of this compound, how much light was getting absorbed between the lens and the vitreous. And the answer is all of it. So this is the path length of the light and how much is gone. And basically, by the time we get to the point of the retina, 99.9% of the light has been filtered out. So the compound itself is acting as its own sunglasses. This is a problem for us. We have to come up with an engineering solution now to get this on the retina but out of the rest of the eye. And we're thinking about ways to do that. We tried injecting it underneath the retina. That was not very successful. We detached a number of retinas but didn't really get any photoswitching. We've tried injecting it behind the eye, hoping it would go through the sclera the same way and get into the retina but not into the vitreous. That didn't work. So we're still working on this.
Give them new lenses. Give them glass lenses.
So in humans, of course, that's what we're going to do. We love to do cataract surgery.
Yeah, I know.
We love to do vitrectomy surgery.
I'm signing up.
So, but the FDA kind of wants to see that this works in animals before we can get to people. So we're going into rabbits is the short answer, where we can take the lenses out. You can't really do cataract surgery on a mouse. They're just too small. But you can on a rabbit, and there is a strain of retinal degenerate rabbits that they have in Utah that we're acquiring now. So to wrap it up, Red Dad, our third generation compound, has some good properties. It's more potent. The kinetics are more normal with the on and off responses. It's very soluble, and it looks like it centers on the bipolar cells, which is terrific. But the problems, it might be too soluble, it stains the lens, it's filtering the light, and as we'll come back to here at the very end, this is what we're toying with, by the way, for delivery, is to make a little pellet delivery device that if we can get it to kind of secrete in one direction, we could potentially get a gradient going where it's much higher at the retina than it is in the rest of the eye. So the challenge for us now is to figure out, now that we know we can turn things on and off, can we make vision out of this? Because it's still not that natural action potential train that happens in the optic nerve. We take a lot of solace in the whole cochlear implant field, because that was the same problem. You're not really re-encoding the normal eighth nerve activity when you put a cochlear implant in, but the brain figures out the temporal relationships and creates something like hearing. We would be very happy if we got something like vision. We know we're not going to get normal vision, right? There's no way we can do color encoding, for example. So things are going to be psychedelic. There's no way around that. The anatomic mapping is not perfect with the ganglion cells. So the fovea, remember, has photoreceptors, but the ganglion cells are displaced. So there's no ganglion cells right in the middle of the fovea. So when you look straight on at something with DNAC or Phenac, what you're going to see is a collapse of the center and a perfect little center scotoma, and everything right around it's going to get donut holed in. So we don't pretend that an injection of this is going to bring back 20/20 normal vision. But can we get something close enough that someone could ambulate or read large print or things of that sort? That's really what we're going for. A lot of these other issues could be fixed in software, right? Because if you could scan a raster across the retina, you could make up for those spatial issues. Is it fast enough? I didn't show you our kinetic data. Certainly not as fast as normal cells, because you're blocking potassium channels and that takes a little while, the latencies are on the order of 500 milliseconds as opposed to less than 100 milliseconds for our own retinas. Are all classes of RGCs equivalently activated? We don't know that yet. Can we improve the delivery, as I mentioned, and is there toxicity from blocking potassium channels long term? And it wouldn't surprise me if there weren't. Nonetheless, we've now shown that the photoswitch compounds do confer light sensitivity on genetically blind mammalian retinas in vitro, that this sensitivity can partially restore pupillary light response, so it seems functional, that the newer compounds are better in terms of their white light performance, their kinetics, their dark off activity, that we may be getting to a point where we can actually target upstream neurons, which will give us some processing. And we have a very clear pathway now to translation, which is we got to figure out a way to deliver this. We have to do toxicity studies, and we have to get this into some people. And we just got a large R24 grant from the NEI to do this, which is wonderful. So we're in year 1 of that grant, and our goal by year 5 is to have a clinical trial up and running. So we have a lot of toxicology work to do and delivery work. Lots and lots of people contributed to all this work. From my lab, I want to particularly highlight Joan Nemmergut, who is here, former postdoc in the lab, now in Montreal. Kuldeep Kaur, who's a current postdoc in the lab. Mike Mnookin, who was a postdoc in the department and now has joined our faculty. And Jack Saichev, who did the pupillary light responses, was a medical student at the time and is now a resident in ophthalmology with us. Rich Kramer's lab, John Flannery, and Udi Isikoff at Berkeley are all in our Nanotechnology Discovery Center. And Dirk Trauner is the chemist who made all this happen at UC Berkeley and then at Munich. We're fortunate to have had one of these roadmap grants to support this work from the NIH and more recently the NEI R24, and we're supported as well by Research to Prevent Blindness. So I thank you all for your attention and happy to take some questions. I'm supposed to do this, right, to see if there are questions. So. No questions yet. Okay.
So I love this story, Russ, and it's just so filled with cleverness and you know, happenstance. Bumping into this chemist guy sounds like that nothing would have happened without that. And I'm also optimistic about moving to the rabbit and seeing whether you might be able to outfit someone with blindness with a lens and use this compound. In thinking about this, I'm turning the picture around and looking more at the brain. What's happening with the idea that you've got all this beautifully orchestrated machinery for the interpretation of your visual world? And we do deep brain stimulation. Why not have something back here that's hooked up to a really good camera in front?
Yeah, there have been people who've toyed with that. I think it's conceivable and doable. The problem is we really don't understand the cortex well enough, I think, to do that in terms of exactly where you put those electrodes, how you recapitulate space. You know, the nice thing in the retina is that our retinotopic mapping is really simple. Take a spot in space, rotate it 180 degrees, you get the spot on the retina, right? Not so simple in the cortex, because you've broken your left and right visual fields apart, obviously. You have these ocular dominance columns, which are encoding your stereo specificity, your direction selectivity, all the Hubel and Weasel stuff. So it's not obvious to me that it's easier to do a cortical stimulator and recapitulate a visual world than it is to do a retinal stimulator and let all that beautiful processing machinery do what it's supposed to do. Very hand-wavy answer. There hasn't been a lot of progress on the cortical stimulation front,
I was
though.
just going to ask if anyone is even trying.
People have tried it, but they really haven't gotten a whole lot of result yet. In fact, probably the most interesting sort of cortical thing that has gotten to humans is the visual lollipop. I don't know if you've come across this. So some very enterprising low vision specialist in Canada came up with a little electrical lollipop. You put this thing on your tongue, and a video camera feeds into it, and basically just stimulates the electrodes as if it were your visual scene. And it turns out that your tongue does a pretty good job of creating a retinotopic map. Now, whether it co-ops visual cortex or not, I don't think has been established. But people can actually ambulate around a room with this little lollipop in their mouth.
Yeah.
You know.
Well, that's what I was imagining might be possible because of the incredible plasticity. I mean,
right.
You guys, the reverse world lenses, 3 days, 2 days.
Right, you're
Some
functional
people,
again, exactly.
everything's perfectly fine.
So exactly, I think it's we are counting on plasticity to make this work.
Yeah.
Because even with this approach, where the retinotopic map is intact, because I think that's the hardest thing to get, is if a point in space is really not here and it's not recognizable as a flipped image. If it's all messed up and your nose is over here and your mouth is over here, it's going to be very hard to make sense of the world. But because that mapping stays intact, it's really just making sense of the action potential trains for their kinetics, not so much their spatial orientation. And that's why we like the idea of using the retina and the circuitry that's left to try to restore function. And most of the approaches, again, stem cell, optogenetics, the chips have all really focused on the retina. It's very amenable to this. The problem is we can't use this approach, obviously, for glaucoma, for optic neuropathy, optic neuritis. If the nerve's dead, then all bets are off for these approaches.
What about macular degeneration?
Macular degeneration should work very well for this because it's the same problem of the photoreceptors degenerating. The only question is whether the upregulation of the potassium channels that we see in retinized pigmentosa in multiple animal models happens with macular degeneration. Problem is there is no animal model for macular degeneration. Best we can do is laser large parts of monkey retinas and ablate the outer retina that way. So I think that's going to wait till people. And we're aiming for retinized pigmentosa first, just because that's sort of the more severe
So is it
form.
odd that old animals don't get macular degeneration?
Well, they don't have a macula. That's probably part of the reason. The only foveated animals are primates and birds of prey. So, and birds of prey actually have two phobias, so they're a little different than us.
But
And they have.
old monkeys don't get it?
There have been reports of old world monkeys having drusen, but not the full degeneration that we see in people. And again, that may have to do with the genetic background, that if they don't have the complement factor H predisposition, or J. Maureen Knights actually have an interesting hypothesis that cone alleles contribute to degeneration, and many monkeys are dichromats. New World monkeys are all dichromats, so they're lacking one of the cone alleles. But no, there's no naturally occurring monkey model of
I was sitting
macular
right in the
degeneration.
same chair a few days ago. We were having the same discussion about whether old monkeys got Alzheimer's disease.
And do they?
Nobody's sure.
Yeah, with the macular degeneration, it's a little easier because you can just look and tell. And to my knowledge, there is no colony of monkeys out there that is sure to get macular degeneration.
So can't your better world chemists come up with a substance you can inject into the vitreous that lets light through, but doesn't allow that chemical to come back into it?
Yeah, the problem is that that wavelength, that yellow, is what makes it magical. It's a dye essentially, and that yellow color is what you see when it's in its cis conformation. So there's no way to take the color out of it and still have it work, unfortunately. So I think the solution is not so much chemical, but engineering. And what we've been talking with a group at Hopkins is they do, I think it's called electrostatic spray technology. They can take a piece of polymer, like a plastic polymer, acrylamide, or acrylic, and they can spray on a drug that will time release over time from one side of that compound. So our thought is if we go in and take the vitreous out, take all the jelly out, and then take sort of a transparent semicircle of the acrylic, which is not too dissimilar from an intraocular lens, and spray the back side of it with the drug, and put that right up on the retina as an insert. Then we've decreased the path length to whatever the distance is between the acrylic and the retina, which we could probably get down to well under a millimeter. And in that path length, then less of the light would get absorbed, as opposed to having to go through the entire vitreous cavity, which is 20 millimeters long in some people. So that's the approach that we're looking at, but that's early days. We haven't even put this on the acrylic yet. We just identified a lab that thinks they can do that chemically. It's actually a very tough problem. That's going to be our biggest issue, is solving that engineering, I think.
Russ, as I'm thinking about this, I do think that the brain has enormous plastic capacity that's almost never called on. In some ways, it's currently unexplored. So that's a trump card that you may have without even realizing. But I'm also I'm in awe of the complexity of the retina, and it doesn't seem to get less complex with evolution. It seems to get slightly more complex. So as you pointed out from the beginning, there's a huge amount of information processing that goes on at that level.
It is, and I in turn am in double awe of the complexity of the brain, because I can't even understand the retina, and that's only 3 cell layers thick and 2 millimeters across, and we still haven't figured that out or come close to it. We made a strong push to be a larger part of the brain initiative and the brain mapping initiative. The retina was, I think, a little bit of a forgotten cousin in that, even though it really ought to be the first place to do a complete structure function, because we theoretically...
Spoken like a true chair of ophthalmology.
Absolutely, but my voice did not carry in that debate. But I agree with you. If we can't understand how the retina processes, it's going to be very difficult to understand many other parts of the brain where the connectivity is so much more complex and the functional relevance of the cell types is that much more complex.
But I like your approach. I don't know how aspirin works, but I'm perfectly happy that it does.
This is translation. When people say, well, what do you mean by translational research? This is translational research, right? We have good, solid chemistry. We know some basic neurophysiology. We understand how our compounds work. But now the trick is, can we make it work towards a clinical end? And when everyone talks about translational research, this is what I envision it meaning. Because you can't do this without understanding the basic science. It's not like we're just randomly trying stuff to see if we inject 50,000 compounds is something going to restore vision. But the flip side is this is not basic science. And if this went up, and it has, for an R01 study section, it just got destroyed as not hypothesis driven, et cetera. So thankfully in the NEI there is an R24 mechanism, which is exactly a translational science large grant. It's like a P30 or a PN2 type U10 multi-investigator larger grant to support this type of work specifically. There aren't many of them, but we were fortunate to get one of them.
I have a question for you. So this extra sort of floating DAD in the solution, creates what we call the inner filter effect. So for a chemist like me.
Mm-hmm.
So I just wonder, are you guys thinking any work about how to get a more specific targeting group than triethylene? Because that
So Dirk has derivatized this unbelievably, and he has a freezer full of interesting compounds, glutamate receptors that are marked. There's a whole approach, and if you're interested in the papers, this is Udi or Ehud Isakov's group, has a 2-component photoswitch where they made a genetically modified metabotropic glutamate receptor and then hooked it to a azobenzene modified glutamate with a maleimide at the end. So they call that MAG, maleimide azobenzene glutamate. And it works really well. And they get signal amplification because it's metabotropic. It's not ionotropic. So Dirk has a huge number of more specific approaches. The challenge for us is that chemically it's hard to do that. There are very few things that as a direct chemical will be highly specific for one cell type in the retina. So the one thing that we thought we might be able to do was there's a particular metabotropic glutamate receptor that's of high interest to retinologists, which is the mGluR6, the number 6 receptor, because it's only found on the off pathway of the retina. So if you just stimulated that, you would really just get one bipolar activation pathway. And we had another chemist, Phil Sullivan, who worked on this a little bit. He's in Montana now. And he tried to derivatize there are a couple specific mGluR6 ligands, and none of them worked sterically when you put an azobenzene on them. And I think he tried another group, a hemethiol, some other group that also photoisomerized, and that didn't work either. So we got very lucky, I think, that the quaternary ammonium worked in one conformation. Sterically, it could have destroyed, all activity from the compound. We just got very lucky. We got double lucky, actually, because we first tested it on blind retina before we went back to the wild type. If we'd started on wild type, we would have given up and just said this stuff doesn't work at all. So there is definitely some hit or miss in that. But I agree, ultimately, it would be great to target this. We'd just like to target it without gene therapy and without having to do an introduction of a mutant receptor, because then there's not much difference between this and just doing optogenetics.
I'm not a channel guy, but is there a specific type of potassium channel you're deeming with?
TEA is pretty non-specific for the whole KV1.X family. So early on, Rich Kramer took AAQ and tested it against heterologously expressed KV 1.1, 1.2, 1. Up to 1.8, and it worked to varying degrees against all of them, which is what would be expected because tetraethylammonium is a very non-specific potassium channel blocker. One of the compounds, which I didn't show you, is QAQ, which has the quaternary ammonium on both sides and isomerizes, and that one, very interestingly, blocks sodium channels. And when we look carefully at the structure, it turns out that it's almost a dead ringer for lidocaine. Really interesting. So effectively, he made a light-dependent lidocaine. We thought for a long time, how could this be useful? Turns out it's really not. If you want lidocaine, you want it all the time. You don't want to turn off the lidocaine. So we couldn't think of a good use for that.
Turn the light back on.
Yeah, exactly. Exactly. It's you'll be pain-free until you go to sleep tonight, and it's going to hurt like hell, right? So it really doesn't work clinically. But these are very non-specific blockers that we're using right now, and I think there's a lot of opportunity to hook this to other drugs.
But is it known the channel in the ganglion cells or the bipolar cells that it's working on? What type of?
Yeah. So we have a little internal debate in our group about this. So Rich published paper in Neuron last year, and I'm a co-author on that paper. Although we had a long discussion about the data on this, where he felt that it was a particular channel called the HCN channel that was upregulated in degenerated retinas and was responsible for this effect. And there's no question the HCN channel is upregulated in degenerated retinas, and there's also no question that AAQ or DNAC is active against the HCN channel. The problem is that the data that are in the paper showing that if you block this channel pharmacologically, you lose the light effect. What we see when we block the channel is very broad, non-specific retinal dysfunction. The retina just kind of falls apart under HCN blockade. So I think that the experiment, although the data are correct, I think the interpretation is incorrect. I think the blockage of DNAC activity by HCN channel blockers, competitive blockers, is a non-specific effect, because I don't think we could see any real light-dependent effects in an HCN blocked retina. It's just very essential to normal retinal function. That's the best guess we have right now. I suspect that this is hitting many receptors, though. I don't think it's a single subtype, just based on how promiscuous the compound is when you do heterologous expression and how many potassium channels are expressed in the retina.
Sometimes it's good to be indiscriminate.
This is true. It's better to be lucky than good, right? That's my lab's motto. So.
Russ, that was absolutely terrific, and we're going to get you back.
Well, happy to be here, and happy to bring Coles to Newcastle. So anytime.